When you hear Sanger sequencing, you probably think about DNA. But Frederick Sanger was actually, his first methods for sequencing were for proteins. And he determined the first protein sequence, that of the hormone insulin. So how did he do it? Basically, he invented this reagent, so like this chemical that could label the N terminus of protein. So proteins are strings of amino acids, or like protein letters. And one end they have the N terminus, and then at the other end they have a C terminus. And he had this reagent, made this chemical that would specifically attack the N terminus and lysine, as we'll talk about. But anyway, so we could label the N's. Then with acid hydrolysis, he could basically chop up these different letters, so split the letters apart. And then use methods like chromatography methods to basically separate these basic pieces. So kind of like, where you might have done in like grade school, where you have draw those lines of paper with markers, and then you stick it in like a solvent, so some like water or something, and then watch the different colors in the ink separate. So we did that sort of thing, but he actually did it in like multiple directions with a bunch of different solvents to really separate these amino acids. And because one of them was labeled, that would tell him which was the end. And then because he had all of the, he was separated all of them, he could tell what other letters were there. And so this gave him the end letter and what other letters there were. But what he wanted to know, there was the sequence of the rest of the letters too, right? So in order to do this, he took that initial one and he cut it into pieces. Not individual pieces like we did before, but larger pieces. And then he took these larger pieces and he labeled the ends of each of these pieces. So using that same reagent, that same chemical, he labeled the ends of these. And then he split these up and did the same thing to see what was in each of these pieces. So then for each of these pieces though, because he'd separated them first, he knew which was the end of that piece and he knew what was in those pieces. Then he could cut them up in different ways, and this would give him overlapping pieces. When he did this with cutting them in different ways, he'd have overlapping pieces, and for each of those pieces he would know the end and what else was in those pieces. And this would allow him to piece together the sequence of insulin. So let's look in a little more detail.
Insulin, we've talked about it before. It's this hormone, so like a chemical messenger, and it helps tell cells to let in glucose. And so to let in sugar. And so with people with diabetes, they either don't make enough insulin or their cells are resistant to insulin. And so the glucose can build up in the bloodstream and cause problems and not get used properly. So basically insulin though is made as this like one long chain of amino acids. So amino acids are protein letters. So each of these little circles is representing a protein letter or an amino acid. Sanger was figuring out what we call the primary structure of insulin, the order of its amino acids. So amino acids are protein letters. They have this generic backbone part, which has an amino group. So like a nitrogen with bound to hydrogens and a carboxyl group. So it can be a carboxylic acid, and then at higher pHs, it deprotonates into a carboxylate. So basically, you might see it in either of these different forms, and same with like different protonation states to this. But you have this amino group and this carboxyl group, and so all amino acids have this, and it allows them to link up. And when they link up, they use those groups, so that you're only left with this one amino group at one end and a carboxyl group at the other. And so we call this one with the amino group the N terminus, and then the one with the carboxyl group is the C terminus. And so you can link a ton of amino acids in a row to get a polypeptide. And so of course, though, although they have the generic part, there are about 20 different common amino acids, and they each has a unique side chain or an R group that sticks off. And so these R groups have different properties. And because different proteins are going to have different chain links and different orders of letters in those chains, that's going to affect how the protein folds up. So we call the order of amino acids their primary structure. And then you get secondary and tertiary and quaternary structure. Basically, the structure is all like when the protein folds up. And so when we talk about Dorothy Hodgkin in an earlier post, I was talking about how she discovered like the tertiary and quaternary structure. So quaternary is just where you have like multiple copies of something working together, like multiple chains of proteins combined. And so what's confusing with insulin, as we'll see, is that it can actually form these like hexamers, but even the monomer form. So even the single chain is actually 2 chains because those chains get cut, but it was made as a single chain. So it's still called a monomer. But because this sequence of amino acids is really important, so like the order of the R's is really important because they all have those different properties. And so it's going to influence how the protein folds up in order to like best accommodate their different properties, and like whether they're like water, whether they're charged, all of those different things. And so the order of amino acids is really important in order to determine, like, in order for the protein to make its final structure. I mean, this order of amino acids, so this primary structure that Sanger wanted to figure out for insulin. So Sanger's carrying this work out in like the 40s to 50s. And it's kind of ironic that so insulin is kind of like a tricky protein. So you'll see this with both Sanger and Hodgkin's, that it seems like insulin should be an easy protein. It's nice and small, but it turns out that it has these features that make it challenging. So for Dorothy Hodgkin's, who figured out like the 3D structure, it turns out that it can form, although the active form is the monomer, so like a single insulin protein, it actually can dimerize and then actually hexamerize, so like 6 copies working together, so all sorts of confusion. And for Sanger, it turns out that it has, although this is all made as a single long chain, it actually gets cut up and you end up with 2 chains linked together through these disulfide bridges. And it's made in this long chain. And then it actually, as it's getting made, so as it's being translated, those amino acids are being linked together. This end part's coming out first, and this is the signal peptide. And it's like, hey, you need to take me to the ER. And the ER is the endoplasmic reticulum in this case. And so that's where some proteins go that need like further processing and that sort of thing. And so the signal peptide is coming out first. It gets the rest of it all taken to the ER. And so it finishes its translation in the ER. And then the signal peptide gets cut off because you don't need it once you're already like been taken to the ER. And so now you have proinsulin. Then another part is going to get cut out, and you're going to get another cut. And basically this is going to, since you're cutting out the middle part, so now you're left with these 2 shorter chains, but it's okay because these chains are going to stay stuck together through these disulfide crosslinks. So I'll talk more about these later, but these are basically strong bonds. And so these are going to stay stuck together. And so your mature insulin is actually 2 chains linked together through these crosslinks. And of course, Sanger doesn't know any of this at the time. He doesn't know any of this. And he's setting out to figure out like what the sequence is. And so he starts by figuring out this reagent or this chemical that can react in order to label the N-terminus. And then he was able to, so this allowed him to find the ends, then figure out a little bit around where those ends were. And you see, it looks like there's 3 ends that he's labeled, but there was really only 2 N-terminuses, N-termini. But basically this is a C-terminus, but he was able to label this lysine because the reagent or the chemical that he developed, it can label both an N-terminus or a lysine for reasons I'll get into, because they both have like an amino group. And then he was able to figure out the sequence of the whole B chain, the whole A chain, and then how they were linked together. The fact that he found that there were 2 ends was important because it said that there were 2 chains, or at least 2 chains, because there could theoretically have been more chains that also started with the same letter, but based on like the ratios and that sort of thing, So he was able to figure out that there are these two chains. And so that's really important. So overall, this process took 10 years, so from like 1945 to 1955. And so it all started with developing that reagent and figuring out the ends. And I should say that although it took Sanger 45 to 55, so actually in 1935, so 10 years before then, Jensen and Evans had figured out that there was an N-terminal phenylalanine using a different method. And so in his Nobel Prize lecture, Sanger credits them with the first discovery of an amino acid in a protein. And so I highly encourage you to check that out. Developing this reagent was going to allow him to do more, to build upon it. And so I want to tell you more about all of that and how it worked. So Sanger developed this reagent, or this chemical that can react with something. And so he developed this chemical 1,fluoro,2,4-dinitrobenzene, or FDNB, which can react with an N-terminus of any peptide, or the amino group of lysine anywhere in the peptide, to form this DNP labeled amino acid. So basically, when you have an amino group, when you have a primary amino group, so basically it's a nitrogen that is attached to just a single carbon. Where was that? You have a nitrogen that's attached to a single carbon. We call it a primary amino acid. And the places where you'll find that are in lysine, as well as the N-terminus. And so remember, each chain is only going to have a single N-terminus, but you can also have lysine anywhere in the peptide. And that nitrogen can basically substitute out for this fluorine. So you get hydrofluoric acid and a DNP labeled amino acid. So a dinitrophenol. So basically you still have this dinitryl phenol group. So that's where the DNP comes from. And so basically, yeah, so it's kind of confusing because benzene is like this 6-membered ring. And when we attach that to something, we call it like a phenyl group. So that's why we go from FDNP to DNP. But this DNP is important because for a couple different reasons, it kind of changes the properties of the thing. And so for one, it has this conjugated benzene ring structure, and that's going to absorb light. And so it actually gives this like a yellowish color, which makes it visible. And it also alters the properties. So it makes it more like hydrophobic. So basically it is less like, it makes it more soluble in what we call like organic solvents. So basically for something to dissolve in something, you have a solvent, which is the thing that gets dissolved. Oh, sorry, the thing that does the dissolving, and then the solute, which is the thing that gets dissolved in it. So when we talk about like water soluble things, it means that like if you put that thing in water, every copy of that thing, so if you put like a spoonful of sugar in water, every like sugar particle, every sugar molecule is going to get a full water coat. So different, in order for something to be dissolved, the solvent molecules have to give up some of their solvent interactions in order to form interactions with the solute, so the dissolved thing. And so different molecules will have preferences for different solvents, and different solvents will have preferences for different molecules, because it's a two-way street, remember. And so basically different molecules will dissolve in different solvents, and you can use this as the basis for separating things. And so they could use this property in order to help separate the labeled amino acids from the unlabeled amino acids. And then like compare them to known standards. And so when he did this, he found that he got DNP glycine, DNP phenylalanine, and DNP lysine, but he could tell that it was internal lysine and not an end one. So even though I have it drawn here where there's a H here, when this was part of the chain, so this was labeled while it was part of the chain. While it was part of the chain, that amino group wasn't accessible, and so it wasn't like a primary amino group, and so it couldn't react. And so if you only have this one chain, this one modification of it instead of like two, and other things to make it. So he knew it was an internal lysine and not like an end. So he knows he has these two ends, and based on like the ratio of things, you see that like there's an equal ratio of these two, like saying that there's like these two chains per copy. So it's not like you have two of this and one of this or whatever, like they're in an equal ratio. So suggesting that there's these two chains, and that one of them has lysine. And so of course, like it could be that there were like 4 chains or whatever, and like 2 of them had glycine or whatever, but now we know for sure that there are these 2 chains. And remember that these 2 chains are really starting from this one long chain, but that gets processed, and so you end up with these 2 chains. But we still call this a monomer because it was made from this single long product. So the nomenclature gets a little tricky, but that's where that comes from. Okay, so now he knows what the ends are. So now he wants to expand this method to figure out what bigger pieces are. So basically, the basic idea is that if you take something and you can cut it into smaller pieces, label those pieces, figure out what's on the end of those pieces, and then figure out what's in the rest of those pieces. And then you could, if you have overlapping pieces, you can then figure out all these little pieces and see, put them back together in order to get the big picture. And so he has the way that he can label the ends. So remember, he developed this DNP. And he also has a way that he could separate the different letters, like break the peptide apart and separate the letters. And so this is actually known. So the sequence, like even though the sequence wasn't known, so like the order of the letters wasn't known, the actual composition, like so how many of like, or at least like what percentage was alanine versus phenylalanine versus tyrosine versus tryptophan, you know. And the reason is because there are these methods in which you can so basically, you can use acid hydrolysis to cut apart all of the letters. So I talked in a post the other day about how the reaction, like how the peptide bond formation occurs in your body, isn't actually like a direct condensation or dehydration between 2 amino acids, but you're losing the equivalent of a water molecule. So you can see that when these come together, you're losing this and a hydrogen here, so you're losing a water. And so basically you can reverse this by adding water, and so hydrolysis, using water to cleave the bond. So you can't just use plain old water. I mean, like, well, I mean, it does use plain old water, but under acidic conditions. And so basically you wouldn't want just water, like normal water conditions, to cleave these bonds, or else your protein chains would all be like individual amino acids, which wouldn't be good. But if you use acid, like high amounts of acid, it can actually like protonate this and cause make this reactive, and you get hydrolysis. So these will come back apart into individual amino acids. And then you could use methods in order to separate the pieces, and then see where those pieces are. See what those pieces are like, compare them to known like standards for the different letters. And so the ways that they could separate the various pieces, there's a bunch of different ways. And so it's, they're often like chromatography methods. And so when I've talked about chromatography in the past, I've typically been talking about like protein chromatography in columns, often using this like fancy dancy machine called an ACTA or an FPLC. But the basic concept with chromatography, you don't need these fancy columns or anything. You basically just need 2 things. You need a mobile phase and a stationary phase. And so typically, so you have like a stationary phase, like so the resin in the columns, so like those little beads in the column, or even like a piece of paper. I mean, like paper chromatography, which they use a lot here, or some sort of gel. You have some stationary phase, and then you have a mobile phase. And so the mobile phase, so that's where like the thing that with like the solvent is kind of. And so you have you give a molecule an option. Do you want to hang out with the stationary phase, or do you want to hang out with the mobile phase? So in the case of like this column chromatography, like on the resin, so that would be our stationary phase. So maybe it's like a positively charged resin. And then we have a negatively charged protein. And it's going, the negatively charged protein in the mobile phase is going to get stuck on the stationary phase. Well, like all the rest of the stuff is going, the non-negatively charged things will move through. So basically you have a stationary phase and the mobile phase, and then you're moving your mobile phase through all of the terminology. Like if you're using like, there's like various like reverse phase things. So don't be confused about that. But basically you have these two phases and you're trying to see what my molecule would rather hang out with. So if you have like a piece of paper and you have, you know, you might have done in like grade school, where you have like a marker line on the bottom, and then you try like separate the dye colors as they move up the paper, and you can use different solvents. So the things are dissolving, and to remember, different molecules have different preferences for various solvents, and so you could use that to see, like, so if they like the solvent better, they're going to be moving better with the solvent as it creeps up the paper, and if they like the stationary better, they're not going to move as fast, or they're not going to move as far. And so we're going to see this in, this is a key part. So he's actually going to do this in like two directions. So with, and also with like a bunch of different solvents, because different solvents, different molecules like different solvents better. And so by using different solvents, you can separate based on different properties and separate based on really tiny differences in the properties. Especially if you do it in like multiple directions, then you cut those pieces out and you actually, so you can elute the pieces out and actually further process them and all sorts of things. Another method that he uses is like electrophoresis, which he calls ionophoresis. And so this isn't how he's using it, but just how you might have heard the term in the lab and how I've talked about a lot before is like using gel electrophoresis. In that case, we're setting up this this charge gradient in order to motivate molecules like proteins or nucleic acids through a gel, and we're separating them based on their size, because we actually take measures, well, at least for the proteins, in order to like kind of make them all negatively charged so that they'll actually move. So we kind of negate their natural charge and like mask it so they all or have this uniform charge, and then we'll move and we separate them based on size. But you can also like, so he was using methods of electrophoresis or ionophoresis in order to separate things based on their natural charge. And so like not making the modifications to uniformize them or anything, and not relying on like the relying on their actual just natural charge to get them to move, and then you can separate them based on their charge that way. So basically you have these different chromatography methods and these different electrophoresis methods. He's going to use a combination of methods, lots of different solvents, lots of different things to test. And then you can compare them to known standards in order to see where things are. And so his basic strategy is, so if you just took a peptide, if you just took like insulin and you were to chop it up and then you were trying to run acid hydrolyze it, so to get all of those letters to break apart, and then you try to separate them with something like this, it would be really hard to tell everything apart because you just have so many letters. So he starts off with like further fractionating things. So taking that, like his reaction or whatever, and then taking steps to separate them into kind of like bins. So like positively charged, negatively charged, you can use like coal to kind of like charcoal, or to kind of like extract out the hydrophobic ones. So like the water excluded ones, basically separate things based on differences in their charge. And so you can see here, there's like, he used ionophoresis to separate things based on their charge, the charcoal in order to separate things that were more hydrophobic. This to separate like the acidic from the neutral and basic all before that. So basically all these various methods. For cysteine reacting things, which will come in handy later, or cysteine containing things, which will come in handy later, you can actually separate them, because under acid hydrolysis, they're going to get oxidized to this like cystic acid form. And this is negatively charged, even at really low pHs. And so this is going to allow you to separate them from like aspartic acid, glutamic acid. And so this is just an overview of some of like the various strategies that he's using in order to separate these pieces. But he's not just going straight to the acid hydrolysis. Remember, he wants to do like a partial hydrolysis first. So well, it could be with acid hydrolysis or with proteases, peptidases. But so we can take them, so cut them into peptides, or like, so partial hydrolysis with acid, so using maybe not as much acid or not for as long a time. So you're not cleaving them all into individual amino acids, but you're getting kind of like a random selection of pieces. And then he was separating them with these solvents in this like paper chromatography method. So first he would separate in one solvent, and then he would turn his paper and stick it in and do it in a second solvent. And this would allow him to separate the peptides. And then from here, now he would want to fully hydrolyze them and separate the individual letters. And so in order to actually get the peptides out of here to do this further step, there's this like cool contraption to elute the peptides from the filter paper. Pretty awesome. But anyway, and then fully hydrolyze them and separate the individual letters. So in order to actually see these letters, he used this thing called ninhydrin. So ninhydrin is like this dye that'll react with amino acids and form this extensive conjugated system. Basically, conjugation is where you have like all of this. I mean, you see there's like these alternating single double bonds. And this is really good for like absorbing light. And so you get this purple product. So this is actually used for like, was used for like fingerprinting and all sorts of cool stuff. But anyway, going back. So basically it's just this method where you can label amino acids, or you can label things with amino groups. But the DMP reagent that he developed, so that end labeler, it's actually blocks the, because it's kind of, I got here first type of thing, it's going to block the labeling by the ninhydrin. So if you take your sample, and so you take the peptides, and before you hydrolyze them, you treat them with DMP. You treat part of them with DMP, and then you run with DMP and without DMP. When you have DMP, when you have the labeling, you're going to have a band that wasn't there before, or when you won't have a band that is there in the like control, because the DMP labeled one is not going to get ninhydrin labeled. So you'll see that spot kind of like disappear. And this will tell you what was the first amino acid in that chain, in that like that peptide, so that part of the chain. And then in order for that all to be useful, you need to have long chains of overlapping, but he started out with just finding like short chains. So remember, because you have the DMP label, it changes the properties and makes it more like soluble in organic solvents. You can separate it from like the non-modified things. So if he did like a partial acid hydrolysis to break up the smaller peptides, but he but if he did this after, so he starts with the labeling while it's still intact, and then he does the partial acid hydrolysis. This will let you isolate the pieces that were like around, so the labeled thing, and then the pieces that were like around it, the few pieces that are around it. And so when he did this, he could figure out the ends of the chains as well as the part, so the N termini, as well as like this C terminus, because or of this chain, because it has the lysine, and remember the lysine will react with the DNP or with the FDNP, which makes it DNP labeled. And so he could isolate these three pieces and then figure out their sequence. And then he wants to go on and extend this even further to get the full thing. But in order to do this, now he needs to switch from doing the labeling after you've already cut it. So not just before you cut it. Because if you do it before you cut it, then you can only get those few pieces. So they use partial acid hydrolysis, but partial acid hydrolysis is like, it's semi-random. There are some places where it's more likely to happen, but basically they couldn't get all the pieces that they needed, and they were getting too short of pieces. They needed the overlapping pieces they needed to be able to deduce like the sequence. So they turned to proteases. And so proteases are like peptide or protein scissors. So some of them are really specific, some not so specific, but they all have like various preferences about what they'd more what they prefer to cut next to. But there are different types of proteases, and since they have slightly different preferences, you can use a combination of proteases in order to get different cut sites and different fragments. So you can see that pepsin, chymotrypsin, trypsin, these are just different proteases, and you can see that they give different pieces. And so with the peptide cleavage, they were able to identify the pieces. And then they were able to put it all together and get this the full coverage. And side note, some people were actually trying to discourage him from using proteases because they were afraid it would like catalyze the reverse reaction as well, because enzymes catalyze or speed up the reaction in both directions, so like forward and reverse. But it turns out that the reaction is basically, once you cut it, it's not going back because the pieces are much more happy separated than apart. Okay, so this was the B chain. The B chain was somewhat easier in that it doesn't have like this intracellular or intramolecular crosslink, but we're going to get to the crosslinks later. But first, next, I need to figure out the sequence of the A chain. So you use similar methods. So partial acid hydrolysis and protease digestion. And so these are basically just all the different pieces that they figured out with these methods, and then they were able to get the complete coverage, including all the overlaps. But they need to figure out those crosslinks. So we know that, so we figured out that there were these 3 crosslinks. 2 of them were actually interchain. So between the chains, one was intrachain. So within cysteines, between cysteines within this single chain. And so these bonds, these disulfide crosslinks, they're stronger than the normal interactions. So normally when the various amino acids of proteins interact with one another, like so between chains, even they're just based on like partial charge attractions and that sort of thing. So they're just attractions. They're not actually like sharing of electrons. And so a covalent bond, like where we have these, we draw the lines out, those are covalent bonds. And that's where you actually have atoms are actually sharing pairs of electrons to form these strong bonds. And so they're not going to easily come apart, which is why we need to do things like add acids or enzymes to try to cut them up or separate them. The bonds between the cysteines, unlike the attractions that normally happen between the amino acid letters. So we're talking about letters, not like between, not the linked up ones, but like if on 2 separate chains. These are normally just like attractions. But with cysteines, you can actually get full crosslinks. So those are sturdy bonds. They're covalent bonds, but they're not quite as strong as the rest of like the covalent bonds making up like the protein backbone and stuff. And so we can get them to separate by reducing them or to form by oxidizing them. And so basically, because it's an oxidation reduction reaction between the two. And so I'm not going to go into that there. Just know that there's a way to separate these. But first, they wanted to do their analysis while it's not separated. And so they did this partial hydrolysis, and it was oxidized in its oxidized form. So they didn't add the reducing agent. So they kept these links in place. Then they were able to isolate the pieces containing the links while those links are still in place. Then they were able to separate these and figure out the sequence. And by doing so, so once they separated them, then they could like add the reducing agent, or actually what they did was they like oxidized it even further to form cystic acid peptides, and so breaks that, and then it gets the figure out their sequence in their normal methods. And so this allowed them to figure out the crosslinks, which was the last piece of the puzzle. So a couple of things to know that if you're going to like go and read the papers as I'm linked to, and which I encourage you to read because I think they're really interesting. So this should actually be GLN instead of gin. But you'll see like glue, and GLN and asp, whatever. Basically, in some cases, like if you use acid hydrolysis, you can't tell the difference between an asparagine and an aspartate, and glutamine and a glutamate, because when you have like the acid hydrolysis conditions, this can get like hydrolyzed off, and so this asparagine turns into, looks like it's aspartate, and the glutamine looks like a glutamate. And so they weren't able to tell those apart with just like their partial acid hydrolysis, or the acid hydrolysis. So they actually, by comparing because the proteases don't like have that problem, they're able to compare the protease samples and the acid hydrolysis samples in order to see which form it was. But then he actually calls, like, we normally indicate glutamine as GLN, but he abbreviated like GLU NH2. So I hope that doesn't confuse you too. And then the other thing that could get confusing is that he didn't actually know how big this thing was in the beginning, or even for the first several years. So it turns out that monomers are the active form, so like a single copy, or single chain. Like, so remember that that has both the B chain and the A chain like stuck together, but it was one original copy when it was made. And so that monomer is the active form, but it can actually dimerize and even hexamerize, and so like 6 of them stuck together. And this caused confusion, and so they thought it was like 12,000 kilodaltons. Well, it's actually closer to 6, and so there's some, you might run into that being confusing in the papers. Yeah, so it turns out that it can actually form these hexamers around zinc, and so more on this in my post on Dorothy Hodgkin, but it forms these hexamers, and this is like kind of like a storage form. And so different forms of insulin, like pharmaceutical insulin, they can make mutations to try to make it like last longer in this hexamer form, or promote the release from this hexamer form in order to control the release rate. And the final note, so Sanger didn't stop there. He went on and applied similar thinking to DNA sequencing, different reagents and everything. But he determined this method where you can kind of figure out the end letter, and have these different length pieces, and then kind of read the sequence off from there. And so we often, I have a post on Sanger sequencing, and it is just like another crucial technique, and he won another Nobel Prize for it. So I hope you found that interesting, because I think it's a cool story.